I really care about how the extracellular matrix, so that's all the proteins and the sugars that surround cells, how they interact with cells to help cells know what to do and where they are. Part of my work is around creating extracellular environments that help instruct cells and to allow us to then investigate the matrices that cells are secreting. I've also developed various technologies to create in vitro environments, and part of that involved making hydrogels. A lot of researchers have, for a number of years now, used animal sources of extracellular matrix. Sometimes you may do an extract from an animal, so typically collagen, for example, is extracted from rats' tails. Otherwise, there's a product which is an extracellular matrix product produced from a tumour which is grown within mice. That extract can then be packaged, put into tubes and then sold, and that's used quite frequently by a number of different labs. Much of what we're trying to do is to replace those animal products. So rather than having to use the animals to source those materials from, we use something which is created synthetically, and we encapsulate the cells in that and work with those in order to make them behave like they do in vivo. Peptide hydrogels we work with are really simple. They're just a string of 8 amino acids that come together when they're at the right concentration and at the right pH to then assemble into a network which then entraps water. So often what we need to do is functionalize the gels a bit to just keep the cells happy, but the cells are then producing their own environment around them, and that helps those cells behave in a way that is more similar to the environment they'd be growing in within the body.
Yeah, one of the things we were interested in doing in developing these peptide hydrogels is being able to independently vary different properties. With a lot of existing hydrogel systems, if you control the composition, so for example, if you put more collagen in there, you're also going to make everything stiffer. It's really designed to be as flexible as possible in terms of allowing you to design exactly the kind of tissue that you want.
As part of the NC3R's funded project, we were working with a number of other labs to try and make sure that the technologies that we were developing here really worked in other people's groups. We could use the Skills and Knowledge Transfer Fund to link up with the Manchester Breast Centre and also with Professor Valerie Spears up in Aberdeen to be able to then work with them to optimise the hydrogels for their particular applications. By providing a way of growing cells in the lab, in a controlled environment, in a reproducible environment, in an environment where we can better predict the outcome, we can also replace the use of animals. So that particularly links to the work that we've done with Professor Anna Gaborska here in Nottingham, because her work was accessing animal-grown tumours, but we can replace that by using the hydrogels in the lab in order to be able to passage and expand the number of those tumour cells that she uses for her assays.
One of the things we've done a lot of work on is trying to develop models where we can predict response to treatments for individual patients by having what we call close to patient cells, and we started working on patient-derived xenografts. So when you put that piece of tissue into the animals, theoretically you've got all those stromal cells around it. You should be maintaining their microenvironment. But we quickly realised that although actually you seem to maintain stroma, all of that stroma is actually coming from the mouse. So all the cells that come into it, but also lots of the molecules that are being laid down in there are derived from the mice as well. That mouse stroma drives things differently. Every tumour is different, every subtype, every individual patient's microenvironment is different, and what we wanted was something that was more tailored. So I think there's more work to be done, and but that's all feasible with the gels. That's the beauty of them, is that that's all feasible.
We were aware that a lot of labs were keen to take up this technology, but really to have the impact that had got us involved with this in the first place, to really reduce the reliance that multiple labs had on the animal-derived matrices, and to reduce the number of animal models of drug development that are used. We then really started thinking about how we could take this technology out. We knew it was highly reproducible. We knew it was something that people were interested in and really were driven to take up these technologies, but how to get that out there. So at that point, we really realised that commercialisation was a sensible way forward.
And Cathy encouraged me to enrol on the DBSRC iGill programme, where I went out and I spoke to potential end users of the technology to find out what it is exactly that they'd be looking for in a hydrogel product. And after speaking to nearly 100 people across 6 countries, I had to present what I'd learned to a panel of experienced entrepreneurs and investors. And based on their feedback, they recommended that we were in fact ready to commercialise this technology through a spin-out. We were one of the few teams that were selected to apply to the Innovate UK follow-on fund, and that will allow us to set up the latest exciting company here that we're calling Cathy Merry. And we're very excited now to have a vehicle to allow us to bring the next generation peptide hydrogels to the wider research community.
Obviously, I'm delighted with where we've got to with the hydrogels project, but I have big dreams for the future. The technologies have developed, and they've developed fantastically quickly over the last few years. So by linking together some of those analytical technologies with the hydrogels, we can then start to really probe deeper into what the role of the matrix is.
We could predict which of the components of that chemotherapy the patients might respond to, although we were able to predict what had happened. It takes us too long to do it for it to benefit the individual patients at the moment. And what I'd really like to be able to do is to do that in a timescale where it's clinically applicable. I think with the peptide hydrogels, we could probably do that.
There are different patterns in different levels of cancer and also in different types of tissues, different types of diseases. And if we can look at that and understand it, then we can reproduce that in the biomaterial. In my independent research, I'm focusing a bit more on biomaterials development, trying to reduce the use of animals in research, in cancer research, but also generally in fibrotic diseases and conditions.
Our main goal is to reduce and eventually replace the use of animal-derived hydrogels. And we believe that our products will offer all kinds of advantages, such as better reproducibility, lower costs, and the fact that they are not animal-derived means that they are more ethical. And not only that, but we can go in and customise them with specific proteins, specific glycans, and create a more human-relevant environment, which will produce more human-relevant data. If you're interested, you can visit our website, which is pettymatrix.com, or you can email me directly. That's john.pitt@pettymatrix.com.